Juan, would you like to introduce our first speaker for today?
Sure, thanks, Sarah. Good morning, everyone. Thanks for joining us to our annual plant breeding symposium at Iowa State University. My name is Juan Panelo. I'm from Argentina, and I'm currently a second-year plant breeding PhD student in Dr. Sara's lab. I'm pleased to introduce our first speaker this morning. Dr. Mark Cooper is a chair of prediction-based group improvement at the University of Queensland in Brisbane, Australia. He is also the Deputy Director of the IRC Center of Excellence for Plant Success in Nature and Agriculture. Some of his current research interests include the understanding of genetic and physiological basis of long-term genetic gains in crops, the design of prediction-based group improvement strategies combining plant breeding and agronomy, the quantitative genetics and physiological basis of yield and yield stability of crops for agriculture, and the genetics and physiological basis of crop growth, development, and yield in water-limited environments. Dr. Cooper held technical leadership roles in research, training, and application of plant breeding methodology across the public and private sectors. His scientific contributions have had global reach through training and mentoring of many early career scientists, creating new understanding in the fields of genetics and plant physiology relevant to agriculture, improving plant breeding methodology, and delivering improved genetic resources for varieties and hybrids, including row-tolerant maize hybrids developed by the deployment of prediction-based methods. Among his vast contributions to the scientific community, Dr. Cooper has published 146 research papers, 37 book chapters, and has been the co-editor of 2 books. His publications have collectively received more than 15,000 citations. Dr. Cooper has given plenary and keynote presentations at more than 50 international and national scientific meetings. Today, Dr. Mark Cooper will be delivering his presentation named Rethinking the Breeder's Equation, Predicting Selection Trajectories for Yield and Yield Stability. Please join me in welcoming Dr. Mark Cooper.
Well, thank you, Juan. I'll just do a quick check. Can you actually hear me?
Yes.
Yes. Okay. And so I've never used WebEx before, so I apologize, but I'm going to ask questions to make sure I can get this running. Do I use the share option from the top left and share content? Is that right?
Yes.
So then I should be able to select my screen. Pulls up PowerPoint. You can see the PowerPoint?
Yes.
And I'll put it into presenter mode and just confirm that you can actually see that, and the laser pointer options on. Is that correct?
Yes.
Okay, well, I'll begin, and I'll rely on you coaching me if the audio breaks up or I'm not doing a great job. So I'll start now, but thank you very much for that really kind introduction. It sounds like a different Mark Cooper than the Mark Cooper sitting here just after midnight in Brisbane, Australia, but I'll do my best to do justice to the introduction, and really thank you very much for the invitation. Would have really enjoyed to participate in person, but as everybody's explained, we're living through a different dynamic right now. So when I was asked to talk about prediction methodology, one of the things that I thought might be really good to do is sort of give an update on ways I'm thinking about prediction in the context of a breeding program where we're developing new products that can be used, and that builds a foundation for many applications of prediction. But a lot of people are familiar with thinking about a breeding program from the framework of the breeder's equation, and I think most people could understand that we're living in a different world than when the framework for the breeder's equation was originally formed. So it gives us an opportunity to rethink many aspects of how the breeder's equation actually operates, how we might use that, and how we might use it in breeding programs, and certainly from the perspective of plant breeding programs. And the way that I like to think about it is instead of describing trajectories as we've done in the past as outcomes from breeding programs, we're actually in the position to be able to think about predicting future potential trajectories for improving productivity, yield stability, and many other aspects of crop productivity, nutrition, sustainability, and so forth. So what I want to do is begin by making everybody really aware that this is certainly not all just my effort that I'm talking about. It's certainly a collective effort from a lot of people. And I'm just acknowledging main contributors down below, a lot of participants that I work with at the University of Queensland. Here on looking at the screen, the left-hand side, Christine Beveridge is the director of the Centre of Excellence for Plant Success, within which a lot of this work is taking place, and also through partnerships. And one of the partners in this Centre of Excellence is Corteva. And here are some of the participants that have contributed to this work through their activities within and Corteva, some of those in the past and some continue today. So I thought it'd be really worthwhile just recognizing the origins of the breeders' equation. And I think most people at Iowa State University would be very familiar with the contributions from Jay Lush. And for me, I really enjoy looking at the history of a lot of the concepts and thoughts that we take advantage of today. And it's quite interesting to actually go back and read the original publications by Jay Lush, and certainly his book, Animal Breeding Plans. And there's a really nice synopsis of some of the key parts of the breeders' equation we're familiar with, where we think about a genetic improvement from a cycle of breeding. He talked about this very graphically in terms of animal breeding programs, and also through words talked about the selection differential and the concept of obtaining information about the populations that were being improved and extracting variance components and estimating heritability. And certainly it's really worthwhile just understanding the conceptual advances that were going on at the time to form this framework that was well used by animal breeders at the time. And if you read the publications by a number of people that were looking at plant breeding specifically, Haller and Miranda, for example, you'll get a really good sense that this framework wasn't well used by plant breeders initially, but a lot of plant breeders today are spending a lot more time thinking about it. So what I want to do is start off and just step into some of the background and put that concept in action, and then look at some of the advances in the theory that we've been working on, but not actually going into the theory, looking at more putting that into place inside experiments and building methodology to improve prediction as we advance our understanding of the genetic architecture of traits, and some empirical results that motivate how to think about the breeders' equation a bit differently. And hopefully that'll stimulate some ideas and questions and discussion. So we can take Shull's basic idea and actually combine them with another early pioneer in quantitative genetics, Falkner, and his original publication starting back in the 1950s, but his book in the 1960s, the first edition of this, and actually think about the underlying infinitesimal model for a quantitative trait, where you've got a number of loci that are segregating underlying this trait. We can think about a trait that might be controlled by 100 or so genes and have 2 alleles for each of those genes or loci. And we could draw those from a distribution that would allow them to have some expectation of equal effects. And within some reference population, these alternative alleles would have some frequency in that population we can quantify. So these effects actually are associated with the elements that we're trying to select that underpin the variation that we're trying to manipulate through selection to achieve some overall improvement in the mean of that population. So that's the basic concept. And on the right-hand side here, I've just given 2 forms that people would be quite familiar with of the breeders' equation. looking at it in terms of a heritability, or we can think about it in terms of a genetic correlation concept, where we're looking at the correlation between the effects in some reference population to a prediction population that we're interested in, and we can set up experimental situations to look at this. Underlying all of these is this additive genetic variation that exists in the population in relation to this framework. We have these allele frequencies. The important thing that I've been really interested in is the average effect of allele substitution and how that plays out across all these loci. And I'm particularly interested in whether the effects that we're selecting on, whether we can assume these have short, medium, long-term stationary effects, in that the ranking of allele effects might not change in the situations we're working with, and how that might play out. But from this starting framework here, we'll build up. And one of the easy things we can do today, and I'm sure most of the people on this, this symposium would have run some sort of simulation experiments where we could actually take this scenario. We can set up a population with 1,000 individuals, select at some frequency, run a number of replicates of this breeding program, run it for 30 cycles, 100 replicates here, and just look at the overall change in average performance, how the variance components might change as the underlying allele frequencies are changed over these cycles of breeding, how that heritability plays out. And we can actually relate this prediction from the breeder's equation to this incremental effect here, and we can start to think about the underlying trajectories of allele frequencies for each of these loci with these effects under the selection, and think about that within this breeder's equation framework. So today, with our ability to actually sequence the underlying genome of these individuals, we can actually look at the individuals that we're selecting among. We can fingerprint those in different ways. So as well as looking at the phenotypes in an experiment here, this was when I was running a maize breeding program at Pioneer. At one of the locations, we had the early stages of the breeding program in a range of environmental situations here. We could fingerprint all of the individuals that underlie this experiment. And around, what we could do is we could associate the polymorphisms at these loci across the genome with SNP fingerprints, or do that from an IBD perspective on haplotypes with this phenotypic variation. And based on these associations, we could actually start to make predictions for individuals that had been fingerprinted but not yet phenotyped. And when we actually use a framework, there's a number of ways of doing this, such as the methodology introduced by Mawison et al. We could actually start to focus on estimates of what predictive accuracy we could actually achieve. And so here are some early examples where we were predicting into a future year into different individuals for a range of traits that have a range in heritability level, as we think about it in the breeder's equation, whereas now we could actually think about this predictive accuracy, and this gives us a framework for thinking about predicting individuals that have been fingerprinted but yet to be phenotyped. And in this case, this was looking at phenotypes in a future year where the associations were built on data from the phenotypic data and the fingerprints from previous years. So this is a basic prediction methodology that commercial breeding is using today, in commercial maize breeding certainly. And what I'm interested in is going beyond this. And where I would like to think about this is in terms of the short, medium, and long-term outcomes from a breeding program. So here, what I've done is I've just taken experiments that were done on the sequence of hybrids that were released by Pioneer, now Corteva, building on the Don Duvik era experiments where these hybrids were grown in an experiment with each other. And the TPE representation in here is just the traditional Don Duvik type of era experiment where the hybrids were compared in different locations from the Corn Belt. And then we started to test out, well, how was this gain being achieved by evaluating the sequence of hybrids in different environments, particularly looking at water-limited environments and also more favorable environments where radiation might have been more limiting rather than water or nitrogen. So we started to ask questions about this long-term gain, thinking about the improvements that we were making in these cycles currently, thinking about the historical trajectories and what sort of trajectories might be possible into the future, given we were studying variation built on this historical legacy of breeding. And we wanted to start asking questions about, rather than just describing the improvements historically, using a framework of crop growth and development to start to ask questions about which traits were underpinning this. So you could start to think about recasting the basic link function that we use in quantitative genetics, such as the infinitesimal model, around traits that are contributing to growth and development, where they're conditional on each other, influencing growth, development, partitioning, accumulation of yield in different environments. So I'm going to talk about this, the formalization of this as a crop growth model. So it's another sort of gene to phenotype link. Obviously, what we want to do is formalize that. And so here, what I've done is just taken this crop growth model and shown that really it's just a hierarchical organizing framework where we have a series of traits at different levels in the hierarchical framework that are taking these inputs to drive development, accumulation of biomass, and partitioning. And instead of pointing our underlying infinitesimal model to the endpoint trait, we might point it to other traits inside this framework. So our infinitesimal model can start to be thought of through the viewpoint or the lens of this crop growth and development framework, which really starts to focus our thinking through our historical understanding of ecophysiology, agronomy, how crops perform in different environmental situations. So it's really, while it might look a little bit different, it's not that different than the model we have had previously. We're just pointing the underlying infinitesimal model to functions that sit inside the crop growth model that ultimately would allow us to accumulate a yield prediction, but from these underlying traits. And so we can now start to ask some questions about, well, what would these trajectories actually look like if we were looking at the average effects of allele substitution and the selection outcomes through the lens of the effects of these traits through these intermediate traits to ultimately influencing yield as a final outcome. So what that meant was redesigning some of these crop growth models, and this was where a lot of the work with Charlie Messina at Corteva and his team, and Graham Hammer at the University of Queensland and his team, so that we could actually explicitly start to incorporate genetic variation for some of these functions inside the crop growth model. And so we've now got yield as a function of the crop growth model with some traits inside this framework that are these equations that are just the arrows connecting the boxes inside the model. And we're allowing those to vary with the same concepts that we're familiar with in quantitative genetics. So just as an example, one of the traits that we were really interested in, particularly in these drought environments, were traits associated with water use through transpiration. And one of the traits under I guess the definition of limited transpiration that we were looking at was this breakpoint trait that influenced the extent of transpiration in high vapor pressure deficit dry environments. So I'll sort of just walk through a little bit of some of the background experimental work that was actually done. So this is sort of a part of an example of the type of function that we might look at, where the environmental variable here on the axis down here is vapor pressure deficit, the dryness of the atmosphere in kilopascals, and the transpiration rate in terms of millimeters per hour. And if there was no limitation on transpiration, that would just continue as a direct response to the increasing vapor pressure deficit if the stomata didn't close. But if there were limitations, you get some breakpoint where at the extreme there would be no further transpiration under these high VPT environments. if you looked at this over a daily time step where you've got a shift in vapor pressure deficit over the course of a day, these different forms of this function would give you different amounts of transpiration. And so the differences in the area of these curves would be a difference in the amount of water used through transpiration accumulated in this particular day. This is sort of the underlying theory and the function that we're looking at inside the crop growth model, that you could in some way phenotype and map and look at that as a function inside the crop growth model. So the first question is obviously, does variation exist? So we started off working with Tom Sinclair and did a lot of work with his system, which were basically small chambers where we could actually control a lot of the atmospheric conditions in here and measure the transpiration use as we manipulated vapor pressure deficit. And a lot of these early chamber experiments convinced us that in elite hybrids and breeding populations we were working with, we saw variation like this, where there were differences in the amount of transpiration, where some hybrids had no limitation on the transpiration as VPD went up and water was available, but others, even in highly irrigated, no water limitations, would have some upper limitation on transpiration. And that would, in this case, it wouldn't be as extreme as the complete shutoff, but there's a difference in these curves that gives rise to water savings. So then the next question obviously says, well, that's really interesting, but it's pretty crazy. Does it actually happen in the field? So Charlie and his team worked out some interesting systems. So here's an example of one of those where they could take the chambers out into the field, and we could actually do this at a canopy level in the field in experimental plots. And so the same system of measurement, but different types of sensors are actually operating here. And the team would be able to move around a number of these tents around the field. We did a number of these experiments in a lot of different locations to actually measure over the course of a day differences in transpiration rate in different VPD situations. And this is an example from a high VPD environment where we could actually show that there were differences in the level of transpiration that you could measure at a canopy scale level that should give rise to differences in saving of water. And then people would say, well, that's really interesting, but I'm not so sure I'm convinced that that would matter in terms of the amount of water that was available to the crop in the soil. So then obviously we had to do more crazy experiments where we measured in very intense ways differences in water extraction pattern and experiments. And so then we could actually show that there were differences in the overall profile of water use. So in this case, the dashed line here is the one that's actually got no limited transpiration. So it's actually extracting water more rapidly from the soil profile than the one that's actually got the limited transpiration. And this difference around about flowering time translated into about 25 millimeters or an inch of water. So think about that as a rainfall event at flowering time would be the difference in these hybrids of 25 millimeters of or an inch of water at flowering time. And everybody would say, well, that's really interesting, but does that mean anything in terms of yield differences? So then we could go to some of our managed environment experiments where we could have high VPD, we could manipulate these situations, take these types of hybrids and show that in these really high VPD extreme situations, you could get differences as large as 10 tons per hectare because of these differences in water availability for these types of hybrids. And so then we would take this framework, use the crop growth model, and we could parameterize that model and run it over a whole range of situations over many years throughout the US Corn Belt. So this is from like 19, 20 tons per hectare down to somewhere around about this 1.5 tons per hectare. And you'd get this yield difference that would translate into a range of somewhere about maybe 2 tons up to around about 8 to 10 tons in this most extreme situation. But in a lot of situations, you get a small incremental improvement. So it's sort of part of the overall background that we actually had. And so we did a lot of this type of work for all of the different functions inside the crop growth model so that we knew how to set up experiments where we could actually measure these types of traits. And so you can think about then how would you go about phenotyping? So a lot of people are really interested in ways of scaling up phenotyping to understand variation for these and other types of traits. So we could run a whole lot of experiments. So here's an example of one of the experiments I was involved in. So this is all of these points. Each point is an experiment throughout the US, the Corn Belt that we were testing hybrids across. And this is just a fraction of the experiments we ran. But these were the experiments where we thought that a gradient of water was the critical factor to separate the experiments. And all of the green points are experiments where we thought there was very little water limitation. Any red point was where we thought there was a severe water limitation. But there's obviously, this is a very coarse grained view. You could do that at a more fine grained view as well. So we could do this very broad scale type of phenotyping. And we also had these managed stress experiments at specific locations where you could do this more detailed phenotyping. And then you have controlled environment facilities where you can do very, very detailed types of experiments. So the question was, how do we integrate this information? So we got our crop growth model. It started off talking about some of the key traits that we were interested in and looking at variation for. We just talked about one, and I stepped through some of the background experiments. And then we wanted a framework for taking this basic idea of connecting our infinitesimal model to the underlying traits. So we were thinking of yield as a function of a crop growth model with underlying traits in context of environments with some underlying noise variables. And then we, in parallel with the hierarchical structure of the crop growth model, we just set up a hierarchical Bayesian linear model where we've actually got the opportunity to look at variation for these traits and think about the genotyping and the effects of the alleles to these traits through the crop growth model to ultimately influencing yield. And so instead of phenotyping everything, what we were actually doing was using the crop growth model as a link function inside the association part of the genomic prediction. And we're using our phenotyping to start to build up priors and to build these equations inside the crop growth model for ultimately predicting yield. So it sort of put us in an interesting situation where we integrated our phenotyping. We had the machinery for actually implementing the prediction in exactly the same way that people are familiar with it, but now within this hierarchical structure. For all these different types of traits we were interested in, we could also take into account variability and uncertainty in soil parameters that were influencing growth and development. And we could start to ask questions about the average effects of allele substitution, not only just through our straight infinitesimal model, but also through this hierarchical model. So it set us up for doing some interesting experiments where we could compare the types of prediction accuracy that we could get from just phenotyping yield and doing the association between the marker fingerprints and the endpoint traits. Or alternatively, we could compare this integrated framework, the hierarchical prediction framework, to the straight prediction of the endpoint trait in the same experiments. And obviously that took a bit of extra work initially as we were learning how to do this. And this was one of the early experiments where, when I was running a breeding program, where we could set up a range of these water limited environments from water limited low evapotranspiration, low ET environments to fully irrigated experiments, where we could train the model in one environment and try and predict new genotypes into a drought environment, and vice versa. We could train the model in a drought environment to try and predict favorable environments. And so this is the outcome that we actually got from one of these early empirical experiments, where we were working with 4 different families, about 350 hybrids in 2 environments, this well-watered and drought environment, and basically training the model in one situation. So using 3 of these families and trying to predict the left out family into the new environment. And so these are the types of separation that we got, the prediction that we were able to get just from the straight applying Bayes A with the infinitesimal framework to yield on the x-axis compared to the predictive accuracy from the integrated framework. And the really interesting thing is in some of these environments where we were having limited ability to make predictive statements with high skill, we could actually get some lift and improvement using the crop growth model. And underlying this, it was allowing us to take into account some of these shifts through the variation for these traits in the average effect of allele substitution and how that was impacted by the environments to influence the yield predictions. So this was this early experiment that I talked about, and that's published from a special issue of the International Crop Modeling Conference, the paper led by Charlie in 2018. And what I wanted to do was just to introduce you to a more recent publication by Christine Dippenbrock and others, and Charlie Messina and I were involved in this, and Tom Tang, also from Corteva, where now we're doing this at a much larger scale. So exactly the same type of setup, but for a wider range of environments, and now operating at the scale of one of the hierarchical groups of a maize breeding program, and just showing here a comparison for this experiment where we're looking at the predictive accuracy just from the straight infinitesimal model applied to the endpoint trait versus using the crop growth model. And you can see this is the comparison of the prediction among populations. So this is comparison back to here, and there are some of these populations that in this situation they were having trouble predicting into the water limited environments where the crop growth model did a better job. If you actually look at a lot of the within population type situations, you can see that we're in many cases getting an advantage when we're trying to predict into these water limited environments where some of these other traits that are more difficult to associate with yield are actually being handled by the crop growth model. So this is sort of an interesting view, but ultimately what we were interested in understanding was in which types of environments and which situations we were getting this advantage. So instead of just thinking about this as a straight comparison of predictive accuracy, we started to sort out the environments in terms of the level of drought stress. And we could do this by thinking about them in terms of the level of drought measured by some integrative trait, such as the overall crop level, a level of evapotranspiration. So instead of it being drought versus non-drought, it's now on a more continuous scale where the environment's characterized through a lot of integrating a lot of these variables into the overall water that was accessible to the crop with this evapotranspiration gradient. And you can see that where we were getting a lot of this predictive accuracy lift was moving into these environments where water limitations had crept in some form into these experiments. So what I wanted to do was use that as a bit of background and motivation. And what I'm going to wrap up with now is just thinking about this and connecting theory that we were interested in from the beginning right through to some of these applications. So what I've got here is just going back to this original modeling work where this solid line here was from some of our early attempts to actually model maize breeding expectations over cycles of selection, and where we had underlying a wide range of genetic models of the type that I've just shown you, where we went from the straight infinitesimal model right through to allowing different types of interactions, such as to simulating those that are captured by the crop growth model. And this was this solid line is just an expectation of response to selection, running that type of simulation experiment with the crop growth model in place rather than the classical infinitesimal model. And then the solid dashed line up here is the same genetic models, but now we're using some components within that overall genetic architecture where we're saying we're able to capture those like we did through the crop growth model hierarchical prediction approach. And over the course of cycles of selection, you can see early on, you don't see much of an advantage, but cumulatively over time, you can get some sort of advantage over using marker information to get at these underlying components and compare that to just selecting on the endpoint phenotype, or this could be a comparison between the Bayes A approach versus the crop growth model whole genome prediction approach that I showed you. So underlying any one of these differences, each one of these points down here is just a different genetic model with a different G to P architecture. You could think of them as different traits, different types of environments. And I've just sorted these out as a continuum now, where these are the more simple, more additive models on the right-hand side, down to the more complex models on the left-hand side. And if you look at overall these genetic architectures, you can see there's a lot of spread. But on average, in the more complex situations here from this early work, our prediction was that we should be able to get a better lift in these more complex situations if we could capture some of these underlying interactions. So that was sort of a lot of background to the theory, and it sort of motivated this investment into redesigning these crop growth models, looking at whether there were opportunities to use that framework to get at the underlying gene to phenotype knowledge for traits, and being able to deal better with these more complex situations where we're dealing with G by E interaction, epistasis, and other things that people could think about. And what you can see on this continuum, it's sort of an inference or a prediction we're making, was that as the genetic complexity of the trait increases, yeah, it's harder to make progress from selection. But if we could actually capture some of that genetic architecture with the genomic information, the opportunities and benefits from a molecular enhanced breeding strategy you could design should be greater in these more complex situations. But sort of the sting in the tail was that you needed to basically build a greater depth of knowledge of the underlying genetic architecture to enable that prediction. Sort of goes back to some of the comments that Kendall Lamke made at the beginning, was that maybe you've actually got to think about creating the future if you actually want to enable predictions. So you've got to have some sort of framework to actually understand the variation we're dealing with and design the types of products that you want to make for the future. So it sort of puts you onto this approach of trying to build appropriate gene to phenotype models. So that sort of then is sort of this overall framework that I showed you, which really is just a formal way of trying to use a lot more of our prior knowledge inside the prediction process that we're using for making new products in a breeding program. So sort of three final panels just to think about. I sort of just used this basic framework of the breeder's equation, just rethinking how we're actually touching the different components of that when we're operating at the genomic level. And really it sort of forces us to think harder about the not just predictive accuracy, but the underlying factors that influence it, and really think about this right down at the effect of the allele substitution effects across the genome that we're actually impacting. So I sort of tried to build that theoretical framework, introduced some of the methodology that we developed around that, with a framework that was trying to get at the traits and the genetics of those traits and integrating that for prediction. And one of the concepts that I tried to introduce here was that a lot of our investment in detailed phenotyping across different levels could be integrated through this framework, through advancing our understanding and embedding it in the link function inside the crop growth model, but also for using that to build sensible priors that we could actually use to help constrain that and then build that understanding up over cycles of the breeding program. And then I sort of then went from showing how we tried to put that into action and show that this theoretical inference or proposition that we made, we can actually see some of that starting to appear inside some of the empirical data we're working with, where you can think of drought as one of the factors that contributes to this complexity continuum that we're showing in this theoretical picture here. But now we can think about that as drought that's resulting in G by E interaction as we go to sort of more sort of complex situations, in drought situations where other traits are in play, and we have genotype by environment interaction, traits interacting to influence yield outcomes. So it sort of cuts across from the theory through to the methodology, through to the empirical motivations. So just final slide, current themes and future directions. I think through continuing to work with others, there'll be further investments in developing the methodology and improving it for maize applications for the US Corn Belt. And I think if I was to say where we're actually at, it's sort of probably at the same stage as where Jay Lush's first publication on the breeder's equation was. So we're sort of at that really early stages of trying these types of methodologies in practice. One of the other things I'm really interested in is not just how well we can do this for maize in the US Corn Belt, but transferability to other crops and other situations. And one of the living examples of that we're really investing in right now is the transferability to using this for the more challenging environments that sorghum faces in Australia, and then through partnerships with others for other crops and other geographies. I think that there's still got to be a lot of advance in the theory and the methodology for modeling these selection trajectories and applications to crop breeding. And for some of this, we're also looking at how this relates to deeper knowledge across species that we can take advantage of in the evolution of natural systems. So, but I won't talk about that anymore here. But this overall framework, I think, can help us there as well. What we're also interested in is the mechanistic models capture some of the information that we can get at, but we're also looking at the complementarity with some of these machine learning, artificial intelligence methods in partnership. So fusion models are what some people talk about as hybrid models, using crop growth models and these other mechanistic models with these more data-intensive machine learning type of approaches. And so the overall framework is really focused around how to use prior knowledge of genome architecture and gene networks to enhance prediction, and that's sort of a key theme of the new Center of Excellence for Plant Success that we've established at the University of Queensland. And so what I've just tried to do is touch on an example, and I think I'll stop there, hopefully.